are tyrosine 1068, and you can see that the that EGF receptor is phosphorylated under these conditions. Now that means that EGF signaling to the nucleus, as shown by Cohen, is indeed stimulated in the nucleus. And this, then the question is, what is the relation between the EGF receptor and the 120 kilodalton protein, which we think at this time is a derived product from the nuclear native 150 kilodalton PLC gamma 1. And here is that when immunoprecipitate anti-EGFR, immunoblotted with anti-PLC gamma 1, you can see 120 kilodalton. Similarly here and here. And when immunoprecipitate with anti-PLC gamma 1 and immunoblotted with anti-active EGFR receptor, you can see a 170 kilodalton protein. So that shows that the two moiety, 170 kilodalton and 120 kilodalton, which we begin to think is probably a breakdown product of 150 kilodalton. They are these two moieties associate themselves upon both the moieties being stimulated under EGF signaling. Now this raised the question, what are the downstream targets? Downstream targets of the EGF signaling. And the next shows, here it is. You see, the phosphatidylineacetyl kinase enhancer protein, when immunoprecipitate of anti-pike, immunoblotted with anti-PLC gamma 1, what we can see here is the 120 kilodalton protein. And just converse, immunoprecipitate of anti-PLC gamma 1, immunoblotted with anti-pike, you can see here is the 98 kilodalton anti-pike moiety. Now then the next question is, because pike, the other is the PI3 kinase. What is the status of PI3 kinase? And here is immunoprecipitate of anti-P110 antibody, immunoblotted with the anti-PLC gamma, you can see 120 kilodalton protein. That shows that pike and PI3 and PLC gamma, which is 120 kilodalton protein, they form a complex. And if they form a complex, then what is the natural? That there should be an enhanced PI3 kinase activity under the similar paradigm. And you can see here the PI3 kinase activity in 10 and 30 minutes treatment are enhanced. So this support that we have gone through from one stage to the other, and then there is a generation of increase in the activity of the PLC gamma 1, which generates, of course, diacylglycerol and IP3. Of course, there are other mechanisms of diacylglycerol generation, and that we propose that it is the mechanism by which the nuclear IP3 is generated. It is unfortunate that till today there is no way to determine the IP3 in the nucleus quantitatively. We think, and many others think in the same way, that IP3 is generated in the nucleus to mobilize the calcium, but does not stay long enough to be determined by the available techniques. And so the next, then we addressed the question of 120 kilodalton protein. And with the collaboration of B.C. Nair, we have been able to partial sequence 120 kilodalton protein at N terminal and both at C terminal. At N terminal, this is, first 10 is the proline 244 and glutamine 253. And this sequence is identical with the human and rat PLC gamma 1. So this gives us an idea that 120 kilodalton protein fragment represents PLC gamma 1 minus its N terminal 1 to 243 amino acids, and the site of cleavage is located between arginine 243 and proline 244 in the mouse liver PLC gamma 1. This is our current way of thinking. The next, please. Now, so we propose that the 120 kilodalton nuclear protein is derived from the cleavage of 150 kilodalton PLC gamma 1 native nuclear enzyme, and this is based on these facts. First is the immunoprecipitated with isozyme specific PLC gamma 1 antibody. Second is like the 150 PLC gamma 1 is sensitive to isozyme specific blocking peptide. Third, the 10 N terminal amino acids partially sequenced are identical to the one found in the human, rat and bovine PLC gamma 1, and then the C terminal amino acid partial sequence was identical with the rat PLC gamma 1 terminal, C terminal end. And then finally, the immunoprecipitated 120 kilodalton protein fragment displayed phosphatidyl inositol hydrolysis activity. So the next, now this is our final hypothesis on the subject, and we have shown that the IP3 generated by the PLC gamma 1, and this is the IP3 which is responsible for acting on the IP3 receptor located on the inner nuclear membrane and releasing calcium from the nucleoplasm to the from the nuclear membrane, nuclear envelope to the nucleoplasm. I have not discussed the question of how the calcium comes into the nuclear envelope between the two membranes, and this has been done through a number of studies for a long time. First, we identified the presence of the nuclear